Hi, welcome back. This is the first chapter for Unit 3, and we're going to start with Chapter 14, talking about the circulatory system. So we'll focus on blood vessels, blood flow, and blood pressure in this chapter. Your learning goals are outlined for you here. You'll have an overview of structure and function, and key concepts from past chapters and past units include the process of diffusion, osmosis, the function of smooth muscle contraction, pressure and flow rates, which I'll spend some time explaining those here before we dive too far into Chapter 14. So our main focus is going to be learning the difference between an artery versus a vein, and we've got some diagrams to help you visualize those differences. And then regulatory factors that govern blood flow, blood pressure, mean arterial pressure, and cardiac output. So MAP and CO were discussed in the previous unit, and we'll use those terms again here as we start our new unit. Actually, this is Unit 4. I said Unit 3. This is Unit 4. So other systems that come into play as we discuss the properties of blood flow and blood pressure and cardiac output and all these things is a concept from Chapter 11, back in the neural section, Unit 2, the sympathetic nervous system, so the fight or flight response, and also hormonal effects. We'll talk about some of the hormones that we may have touched on in our introduction from Chapter 6. So for circulatory system structures, there's three main things. The heart, which is going to set a pressure gradient in order to pump the blood from point A to point B. Blood vessels that are going to work to distribute blood throughout the body, and those would include arteries, arterioles, capillary beds, venules, and veins, and we'll go through all of those one by one. And then, of course, the blood. Our next chapter, Chapter 15, will be all about blood, but the blood's going to be our transport medium with the capacity to carry oxygen as well as CO2 away from tissues and circulate it back to the lungs so that we can exhale the CO2 off of our bloodstream. And we'll talk a little bit about that, but we'll save blood gases for a later chapter. So the circulatory system has multiple functions, and they're listed for you here. Obviously, the transport of blood gases, so CO2 and oxygen from point A to point B, delivering oxygen to tissues, and of course, getting the CO2 away from them because that would be toxic. And then delivery of nutrients as well as hormones that'll be dissolved in the fluid portion of the blood. Removal of waste, those will be delivered to the kidneys. Temperature regulation, if you think of our planet and how much of the surface is covered by water, our oceans are really what help us regulate temperature on our planet. And because we are mostly water and your blood is mostly water, it helps with temperature regulation. Clotting functions and immune functions, we won't spend as much time on those. Those are very complex properties of blood, and an immunity class, actually you can take a full semester long class on the immune system. So we'll just touch on a few things here. Just know that that is a function, but we're not going to emphasize the processes of clotting. That's like a 48-step process that I'm not going to make you guys memorize. And also the immune function as far as white blood cells or antibodies. We'll touch on those just very briefly. Okay, so structurally, what we're going to do with the circulatory system is to divide it into pulmonary and systemic. So what we mean by the pulmonary circuit, if you look at this diagram here, the heart is set in the middle, and we've got a right and left lung on either side. And the pulmonary circuit handles the blood that's moving between the heart and the lungs. So the heart has to do double duty. It's delivering oxygen to the whole body via the systemic circuit, but the heart also has to pump blood to the respiratory system, which we'll talk about soon in this unit. So the system between the lungs and the heart is under low pressure and low resistance. And if we think about flow, if we think about how water might move um, down a river, it's going to go from an area of high potential energy to an area of low. And that's the same thing that's going to drive fluid through a closed system is going to be an area of high pressure to low pressure. But it's unique in the pulmonary circuit. That's a low pressure, low resistance type of system. And I want to talk a lot more about the physics of fluid flow here as we get into these notes. Okay, so the systemic circuit is responsible for moving the blood from the heart to the whole body. So all of the body's organs and organ systems. The system that feeds the whole body is under high pressure. And that's where the aorta is going to be leaving the left ventricle and starting that flow or that trip through the whole body. So you're going to have very high pressure, and we'll look at the reasons why arteries tend to also set up high resistance. So a little bit of physics, I'll walk you guys through it, a little bit of fluid flow dynamics as well. So pressure is going to be capital P, resistance will be capital R. That's the first key to some of the abbreviations you're going to run into in this note set. Okay, so we looked at some of the circulatory system components earlier, the heart and the blood vessels, and also the blood. So the blood vessels that leave the heart are going to be arteries, and they will branch into smaller and smaller structures called arterioles. Arterioles will connect to capillary beds, and you see the red, purple to blue. Your book uses red to indicate oxygenated blood and blue to indicate venous return, which is oxygen depleted, CO2 rich. The purple, highlighted in the term capillary beds here, indicates gas exchange. So you're going from a rich red blood with oxygen, and then the organs and working tissues are going to absorb that oxygen. So the red turns to blue by going through purple. Capillary beds, which actually feed the organs and tissues, you can see some capillary beds here in each lung on the right and left. And we also, in this diagram up here at the top, we've got the venous return to tissues, and you see the capillary bed here in purple, and also down here at 6 o'clock, other tissues undergoing gas exchange, and you see that purple color. So once the tissues have absorbed nutrients and oxygen and gotten rid of waste and CO2, the capillary beds will feed then to the tiniest veins, which are called venules, and then they become larger veins on that return trip to the heart. So the superior vena cava is the largest vein, and it connects to the right atrium. And then the aorta on the left side of the heart is going to connect to the left ventricle, and that is the largest artery in your heart, and that is a very high pressure artery. The lymphatic system is part of the immune function, part of your innate immune system, and it's going to help absorb excess interstitial fluid. So any of that interstitial fluid can be put back into circulation via lymph, and you can go ahead and flip ahead in your textbook if you have it open and you're already on chapter 14 with me. Figure 14.24 has a diagram similar to this one, but it shows you the points of exchange between interstitial fluid, and that's becoming lymph. So the lymph ducts can help process debris, bacteria, white blood cells will get in there and attack foreign invaders and things like that, keeping your system sterile. The lymph ducts are going to drain into veins, and that processed debris can then be put back into circulation so that the kidneys and the liver can get rid of it. Okay, so Before we move on, I just want to say that the blood vessels that are being classified according to whether or not they carry blood toward the heart or away from the heart. So you can't always go by red or blue. You may have a black and white diagram in front of you. You never know. And when we talk about the size of a blood vessel, when I say large blood vessel or a large artery, I mean the diameter, okay, not necessarily the length. The length is going to be a constant variable, especially in an adult who's not growing anymore. And then also, we want to consider the fact that all blood vessels have a hollow interior called the lumen. That's where the needle is placed during a blood draw. And we will look at that a little more closely as well as we move into the rest of this chapter. Okay, so the physics of blood flow, abbreviated BF. What is the driving force behind the movement of blood from the heart's left ventricle to the entire body? Okay, so recall mean arterial pressure, MAP, okay? We did some measurements in lab, and you had an S1 and an S2. When you were listening through the stethoscope or the modified sounds online, you had sound 1 and sound 2, lub, dub, lub, dub. And when you use the stethoscope to listen and you have a sphygmomanometer, you are able to estimate a person's blood pressure. And you probably got some numbers, you know, on or around 120/80, and that's measured in millimeters of mercury. That's how we measure pressure. So the blood flow to an organ is determined by two things. This would be true for any liquid in a closed system. Okay, so the laws of physics state that flow moves from high pressure to areas of low pressure. Just like when we started talking about concentration gradients across a cell membrane back in Unit 1. Also affecting flow is the amount of resistance. Okay, so fluid can meet resistance during its pathway from the heart to the rest of the body, and that's caused by organs and shrinking diameters of smaller and smaller blood vessels as you move from point A to point B. So the flow is determined by taking the difference in pressure between 2 points, so from point A to point B, and dividing it by resistance. So as resistance increases, if it's in the denominator, the flow is going to be inversely proportional. So as R gets bigger, your flow rate is going to drop. Okay, and we'll talk more about this as well. We know pressure gradients drive the flow. Fluid is going to move from high pressure, P1, say the left ventricle and aorta, okay, over here in the bottom right of this diagram. And it's going to move towards an area of low pressure. So we can go all the way up to the top of this diagram where it makes a U-turn in blue. That's the superior vena cava coming back to the right atrium. And you can see the pressure says 0 millimeters of mercury. So blood flow rates are in units of volume divided by time. So maybe liters per minute or milliliters per minute, something like that. It's directly proportional to the difference between the pressures measured at the two ends of a tube, which would be the blood vessel. And so you would set that up as P1, so 0.1, minus the pressure from 0.2. And the difference between those 2 points is what's going to be often denoted as a delta P. So delta, the Greek letter delta, means difference. And if we want to look at the difference between two numbers, we subtract them. Resistance is going to be anything that hinders flow. It can be friction due to decreased diameter. It can be caused by organs, and even the viscosity of the fluid itself. Lucky for us, in normal human medicine, the viscosity of blood will be a constant. So with pressure gradients, whenever there's a difference in pressure between 2 points, there will be a pressure gradient, and that will drive the fluid from high to low. This is often referred to as bulk flow. We'll talk more about this as well. The heart is your pump, and it sets up a pressure differential. By pumping blood into the aorta on the arterial side, the heart increases the MAP, the mean arterial pressure. MAP is an average pressure in the aorta during the cardiac cycle. This is in the systemic circuit. Since systemic delta P is calculated as 85 millimeters of mercury from the aorta minus 0 millimeters of mercury measured in the vena cava, the average MAP is given as 85. So, reading the concepts that we covered from the previous figure 14.2, the systemic circuit has to withstand high pressures that pulsate with the cardiac cycle. So the aorta, which is the largest vessel in the body, has an average of 85 millimeters of pressure, and that declines over increased distance from the source. Remember, there's a resistance along the way. So organs and then the smaller diameter blood vessels are all going to increase resistance as the pressure decreases and you move away from the heart. By the time blood returns to the right side of the heart by way of the superior vena cava, the pressure is going to be 0. Okay, so gradient blood flow. Let's go ahead and transition into some of the physics involved with flow as we move from the heart through all of the blood vessels in the body. You can see here that the pressure drops from the systemic system as it moves from the heart out into the body. And if you notice, the pulmonary circuit is very, very low. Let's focus more on the systemic, this serious drop that we see in the systemic circuit starting at about 85 millimeters of mercury. And we're going to follow the decrease from different structures as the flow moves through. So if you read the columns along the top, we start with arteries, then arterioles, capillaries in the middle, venules, and veins. And you can see by the time we get to veins, we're going towards 0 if you follow the purple line. So we mentioned before that the pulmonary circuit is going to be very, very low, but we see a more serious drop in pressure. Once we leave the arteries, we get into the smaller diameter arterioles, and we have the most dramatic drop in pressure when we get to the arterioles. And when we get to some structures, I'll show you why that is. And then by the time we get to the delicate capillaries, often diagrammed in purple, when we're feeding the tissues, the pressure is a lot lower. It's going to be on average about 25 in the capillary beds that are feeding organs and tissues from the initial 85 millimeters of mercury where we started. So the arterioles really slow the flow and drop the pressure before you get to those delicate capillaries. And then on the venous side, the capillaries are connected to venules, and then finally the larger veins, and the pressure is very, very low when we get to the venous return. Let's look at the effects of resistance. The pressure drops between circulatory structures. We mentioned this before in the pulmonary circuit that it's going to be low. So the blood vessels, which are acting as tubes connected to a reservoir on either side, there's a pressure gradient. Okay, so the delta P is the pressure gradient, the difference between 2 points. Notice that each tank has the same gradient, but the flow rate is different. You need to ask yourself why that is. So the 2 tanks are connected to each other by blood vessels, and we know that flow is calculated by delta P divided by R. So the bigger the denominator, the smaller the flow. Flow and resistance are inversely related. Blood flow through the pulmonary circuit is going to be the same as the systemic circuit. Flow is going to be about 5 liters per minute at rest. The pulmonary circuit has low resistance due to its physical characteristics, and we'll use veins as an example of this in the next slide. And our rule stated that if the pressure gradient used to drive blood flow through the pulmonary circuit is low, then the resistance must also be low. And the pulmonary circuit has less resistance and smaller pressure gradients, which allows it to achieve the same flow. So here is a great example where you compare the two tubes, A and B, and they give you the flow rate. Well, you can see that B is a lot smaller, okay? It's about half the diameter, which means you have increased resistance, and the flow over here calculated for you on the right is half of the flow rate than the tube above labeled A. So low resistance versus high resistance is going to affect your flow because the two are inversely related to each other. Let's look at some structures and keep in mind the effective resistance that we just talked about. So the structural features that contribute to blood volume and blood flow resistance are shown here. We've already mentioned things like tube length and diameter, as well as fluid viscosity, which is the thickness or the stickiness of the blood. They all collectively affect resistance. But when we compare an artery to a vein, like we have here in figure 14.5, on this cross section, we can easily appreciate the effects of internal diameter. So we are looking down inside the lumen of the blood, where the blood is held and pushed through to all parts of the body. Now, an artery shown here on the left, and your book always uses red to show oxygenated arterial blood flow. There's an obvious smaller internal lumen and a thick middle layer called the tunica media, which has a large amount of smooth muscle in that layer. Arteries are able to constrict by smooth muscle contractions or dilate. That seriously affects the resistance. This middle layer, tunica media, is right here in the middle. And that smooth muscle layer can affect the diameter of the lumen, which is where the blood goes. In this diagram, the artery is connected to the vein over here on the right by a capillary bed. These are not shown in purple in this particular example, but the capillary bed is going to indicate oxygen exchange with the tissues. And we'll talk a lot more about these in a little bit. If we look closely at the vein over here on the right, we can see a large internal diameter in the lumen. And some valves, you can also see this V-shaped valve right here. Okay, those are valves like what we saw in the heart. And just like their function in the heart, valves ensure one-way blood flow. In a vein with low resistance due to large diameter, we can maintain flow under low gradient pressure. So structurally, veins and arteries are very different because they have a different job. Okay, so high pressure, elastic recoil characteristic of an artery, whereas a vein is a large, low resistant type of blood vessel with low pressure returning blood towards the heart. And then in this next figure, we can see the structural characteristics in figure 14.6, I believe this is. It kind of serves as a reference table of the special features for all blood vessels in the circulatory system, as well as the measurements for each structure are given for you here. This should help you grasp the idea of resistance from different sources, but overall blood flow is controlled by the diameter of the lumen. Less resistance means better flow. The smooth muscle in an artery means it will have elastic recoil. So like a rubber band, when stretched, it will accommodate high pressures and then also have the ability to snap back into its original shape. These blood vessels can also constrict, which will shunt blood flow to certain areas when needed. But veins with their thin walls and large diameter lumens that allow these structures to carry large volumes of blood with low resistance. Okay, Okay, now here's a look at the amount of pressure and stretch that the aorta has to undergo during the cardiac cycle. Arterial blood pressure is determined by the aortic pressure. So the left ventricular pressure falls to 0 millimeters of mercury during the cardiac cycle, while arterial pressure stays elevated. Elevated pressure in the arteries during diastole is because of the elastic recoil properties in the arteries. Arterial blood pressure is estimated with a sphygmomanometer and a stethoscope. You can review this procedure in figure 14.8, and we also had a lab simulation on this procedure as well. So the average or normal blood pressure is going to be between 110 over 70 to 120 over 80 millimeters of mercury. And to find the pulse pressure, you're going to take your systolic number, which is a big number on top, minus diastolic, which is the small number on the bottom. And then for MAP, the calculation you can use is systolic plus 2 times the diastolic number. In this case, your example equals 250. So you want to hit equals on your calculator first and then divide by 3, and you get 83.3 millimeters of mercury. That's a very good estimate of our clinical index of 85 millimeters of mercury for mean arterial pressure. Okay, so now that we've seen the structure of arteries versus veins, let's consider the area where the biggest drop in pressure occurs. This will be due to more resistance. Arterioles are the passageway to the capillary beds. These tissues can exchange dissolved gases and solutes across the plasma membranes. So the idea of using a high-pressure fire hose to feed the delicate cells that make up tissues and organs is simply not going to work. From the top of this graph here in figure 14.9, we see the high cyclic pressure coming off the aorta from the left ventricle. And as the arteries get smaller and move away from the heart, the pressure naturally drops. But the steepest drop is in the arterioles. Okay, they provide the greatest resistance. Large arteries become arterioles, and arterioles connect to capillary beds that feed the tissues. If you want to jump over to figure 14.17, you'll see external precapillary sphincter muscles that form rings of smooth muscle that help drop the pressure and the flow rate before getting to the delicate capillaries. But on this diagram, find the column that says capillaries. The pressure averages about 25 millimeters of mercury here, and those capillary beds connect back up to venules, the tiniest veins we have in our bodies. So the pressure is very low when starting the return trip back up to the heart. Venules lead to larger veins. On this graph, the pressure is almost bottomed out. Remember, the veins have very low resistance, and the one-way valves will help keep this low pressure part of the system moving in the right direction towards the heart. Okay, so let's look at intrinsic control of blood flow. And you might be thinking intrinsic control, but actually we've already kind of covered intrinsic, meaning built into the structure. It's got intrinsic control. So the way we control blood flow is by increasing or decreasing resistance. R is our denominator. So all of these structural features indicate that there's an intrinsic method of control. There's a built-in structure function property that helps achieve the goal without damaging delicate tissues. And that's all when that's all going to be when you consider this middle layer. Okay, this middle layer is really well developed on arteries and arterioles. So having that smooth muscle ring allows you to either constrict or dilate, and that will affect blood flow. When we consider blood flow to an organ, we have to just remind ourselves of those changes in the diameter, like what we saw in figure 14.10. And then if we consider blood flow to the organs, this is an illustration from figure 14.11. in several different panels, so you can kind of get the idea that different organs demand more blood flow than others. Like the brain requires a constant source of oxygen and cannot survive more than a few minutes without it. So the carotid arteries come straight off the aorta and run on either side of your neck in order to deliver large quantities of blood at all times to the brain. The kidneys are another example of this. They pull about 20% of the cardiac output by way of the renal arteries, and those come straight off the descending aorta. So some organs just demand more blood flow than others. They're, metabolically active tissues. So the kidneys are another great example of that, just like the brain. So here we have three different organs, A, B, and C. The heart up at the top acts as the pump and sets up the pressure differential, while the organs are all sources of resistance. In red is the arterial pressure, and on the right in blue is the venous return pressure. Your delta P, which drives blood flow through these organs, is equal to the difference between arterial pressure and venous pressure. You can calculate that by setting up a subtraction problem on your calculator. Here we can see the effects of delta P from the organs, indicated by A, B, and C. The flow rate through each of these organs is 1.5, 1.0, and 0.5 liters per minute, respectively. If we consider panel C here at the bottom, organ B has reduced flow, and only organ B. That's due to its increased resistance, most likely caused by the arterioles constricting that lead to that organ in particular. There's also the possibility for neural override. For example, the blood flow to the stomach can be restricted during times of fight or flight. Also, here's an example of how arterioles can react to different metabolic conditions. The smooth muscle layers in the tunica media are sensitive to the interstitial fluid composed, in the surrounding organs and tissues that they supply. So chemical solutes like oxygen, carbon dioxide, potassium, and pH levels can all trigger a vasoactive response, which is what we're seeing here in figure 1412. So for example, when oxygen levels are low and CO2 levels are building up around the tissues, it can cause a vasodilation reaction. For example, when the concentration of oxygen is low and CO2 is building up around working tissues, it causes a vasodilation reaction in order to bring more oxygen to that area. This is often why people look red when they exercise. There's a higher demand for oxygen, and that requires increased blood flow to the area. Hyperemia is a term we use to indicate increased blood flow to an area. And hypoxia is used to indicate decreased oxygenation and often can be accompanied by blue lips or blue fingertips, a buildup of CO2, which is the green set of spheres here in this diagram, can trigger vasodilation. Blood flow to organs is called perfusion, and that has to be maintained at all times in order to maintain homeostasis. Perfusion can be supported in a hospital setting when patients become critical. An example of active hyperemia is shown here in figure 1413. The tissues have an increased metabolic rate, so demands for O2 and blood flow are increased. As consumption goes up, the cardiovascular system has to compensate, and vasodilation in the arterioles is the first to respond to these demands. Vasodilation creates decreased resistance in that particular area and will result in more oxygen getting to that area. There is a negative feedback loop here to maintain homeostasis. So once demand goes down, the system will balance out between carbon dioxide and oxygen, returning to normal levels. So extrinsic regulation of blood flow. Those will be things from outside the blood vessel itself that can affect blood flow. Now let's look at the outside mechanisms that might affect perfusion to an organ or tissue area. Extrinsic control mechanisms will work to maintain MAP, which is the gradient that drives blood flow. When we consider this concept in a global or whole body sense, Flow is driven by cardiac output, which determines mean arterial pressure, or MAP. When we consider MAP and any resistance along the way, we can calculate the total peripheral resistance, or TPR. Another way to think about MAP is to multiply stroke volume times heart rate times TPR. The sympathetic nervous system will override the intrinsic controls we already mentioned. When adrenaline gets dumped into the bloodstream under times of great stress, it will have a global effect. Epinephrine, norepinephrine in an 80-20 ratio secreted from the adrenal medulla is capable of binding to two different types of receptors, the alpha or the beta. When epinephrine or adrenaline binds to the alpha adrenergic receptors on arterioles, when that binds, that will cause vasoconstriction. However, tissues that express the beta version of this receptor, usually in skeletal muscles and cardiac muscles, epinephrine is going to bind. These tissues tend to have a reaction of vasodilation. So you can see why tissue-specific presentation of these different receptors allows for kind of a calculated effect. So if you're going to dump a chemical into the entire bloodstream, the way to manage the effect is through the receptor itself. So some tissues express beta and other tissues express alpha. And skeletal muscles and the heart are definitely going to be a high demand organ during times of great stress. You're going to want lots of blood flow going to the heart and your skeletal muscles in case you have to run away from danger. There's also hormonal effects. Those are considered extrinsic regulation also, since they come from a different source than the blood vessel itself. Vasopressin, or antidiuretic hormone, is one of those hormones that acts on the kidneys to reduce fluid loss. That was mentioned in Chapter 6 as a hormone from the posterior pituitary. You also, we'll talk about it more in this unit when we get to chapters 18 and 19. Another hormone is angiotensin II. We'll spend quite a bit of time on this one. It's stimulated by the release of renin from the kidneys, and that'll be in chapter 19 when we get to the renal system. So vasoconstriction will increase your total peripheral resistance, or TPR, as well as MAP by increasing fluid volume. So we can affect pressure in both ways. Okay, so let's look at some other structural features that add function to these different blood vessels. Let's go back and look at our connection between venules to capillary beds to, arterioles to venules to the capillary beds. In this diagram of microcirculation, you see a lot of branching and surface area. This is what actually feeds the tissues. This is a very delicate, low pressure environment. If we look at these structures highlighted in purple, they're designed for increased surface area due to all that branching, and they're also thin-walled, one membrane thick, and that allows for maximal diffusion. Capillary beds are by design very small with extensive branching, which we see here, and it gives them large surface area. So the top graph over here on the left shows the total cross-sectional area produced by capillary beds, so it gets way, way increased when we get to capillary beds. And then the lower graph indicates low, slow velocity. So the time that we need for diffusion to occur at the tissues is also promoted. We want to have time to exchange with the tissues in this purple region, which is the responsibility of the capillary bed. So structurally, they're very, very small, thin-walled, and they have extensive branching, which helps them be very, very efficient with exchange with the tissues. So here, if we zoom in to the capillary membrane structure, we see how thin it is. This feature, combined with the large surface area, means we can feed the hungry tissues quickly and with great efficiency. And then the small water-soluble molecules, including some fatty acids, and of course steroid hormones, are going to easily diffuse across the membrane structures. And then we also see in the lower diagram a structure called fenestrations, which is a fancy way of saying pores. These will allow for larger substances and particles to cross, like proteins in some cases. Here in figure 14-18, we can see the action of a white blood cell coming out of circulation into tissues. This is a process called diapedesis, and it allows for white blood cells to take care of any infection that might be in the tissue. So the white blood cells definitely need to come out of circulation if there's a loss of integrity in the skin or an infection, things like that. Okay, let's look at capillaries as far as fluid exchange goes. So we looked at some of the cellular processes like diapedesis. Let's look at the fluid exchange across the purple capillary zone. Fluid exchange in the capillaries is going to be driven by Starling's forces across the capillary membrane. So the figures in 14-19 describe the movement of fluids into and out of the capillary beds, as well as from left to right, which is the direction of bulk flow from high pressure on the red side towards the area of low pressure on the blue side. So the pressure found inside the lumen of the purple capillaries is just enough to drive the exchange toward the interstitial compartment, so absorption can occur. These membranes act as a filter. Filtration, by definition, is the process of pushing fluids across a semi-permeable membrane from areas of high pressure to low pressure. This is still considered a passive process and does not require any energy from the cell. As the pressure drops from left to right, 38 millimeters of mercury to 16 on the venule side, the process of filtration and then absorption can occur. So if you can imagine working tissues in the area producing waste, including CO2 building up over time, there's an obvious pathway for solutes and gases to move when dissolved in fluids. Absorption of the newly delivered substances and removal of waste as the capillary flows toward the venule side on the right. So let's look at this with the numbers. Okay, so you can do the math and prove this method to yourself. The net filtration pressure, or NFP, can either be positive or negative. The difference between these two pressures will drive the bulk flow across the capillary beds. Positive net filtration pressure will drive the filtration side, and a negative net filtration pressure will promote absorption of fluids to be returned to the venous blood flow. About 20 liters per day is filtered in this way. Just wait till we talk about this in the kidneys, where we'll be talking about 180 liters per day. You'll get an idea of high demand organs when it comes to blood flow in the coming chapters in this unit.